Namaste everyone. This is Dr. Pratibha K.S. Head, Department of Biotechnology at Kelly Society's S. Nitlingappa College, Rajayanagar, Bengaluru. Today I'm before you to present Industrial Biotechnology Practical, a question number third spotters list. I'm presenting before you all the list of these spotters. Just read those spotters more than enough during this end COVID-19 pandemic situation. Do not panic yourself for too much with confusions. Okay. So here question number three spotters carries 3 marks each, that is identification 1 mark and comment 2 marks. Okay. Before I begin today's presentation, I would like to share this quote, and I want you to take this affirmation on daily basis. Okay. I don't treat failure as an end of the journey. It is only a signpost to take different route. Okay, I will repeat for you. Take this affirmation every day in your day-to-day life. I don't treat failures as an end of the journey. It is only a signpost to take different route. Okay. Before I start today's presentation, I request everybody kindly subscribe my channel to support. This is the spotter. Which one? Yeah, it's given already. Industrial, that is an industrial aerobic fermenter, right? This internal view you can see here. You need to identify this as stir tank fermenter or typical fermenter. All the parameters of the fermenter you can see here, like steam valve, then harvesting zone, cooling water in, cooling jacket, impeller, viewing port, steam, then style seal, motor, pH, pH controller, acid base reservoir and pump, filter, exhaust, cooling water out, baffle, culture broth, and sparger. Okay, a high pressure air will be sent through this particular sparger for the aeration purpose. Okay, here style air will be sent through sparger. Okay, in detail you can also study in theory paper also. Okay, so you identify this spotter as stir tank fermenter, and here I've given the explanation. Kindly take screenshot or you can pause the video, go through all these points. Any 3 to 4 relevant points, if write more than enough. If you know more information, you can write that also. Okay. And also if you're missing any slides, no, please go through this slide number. I've given the slide number here. According to that, you can follow. Hope you are able to recall this spotter. This is airlift fermenter. Okay, you need to identify this spotter as airlift fermenter and comment on this particular airlift fermenter. Write these points. Okay, any 3 points more than enough. And kindly important points you need to underline. That is very, very important to impress the examiner. Okay, and maintain neatness. I hope you are able to recall this. This is, yes, you're correct. Cheese, right? This is the answer. Okay, go through this particular points and make a note of this one. Okay, write 3 points more than enough. If possible, you can write all this and keep ready with you so that you can study for your examination. Okay, then what is this? It's a packed bed bioreactor or packed bed fermenter. You can see here the, right? You can see here the pack, right? So you can keep this in mind as packed bed fermenter. Okay, this is the information description about the packed bed fermenter. Go through this one. Next. Are you able to identify this? Yes, you are correct. This is tubular fermenter. Okay, this is the description about the tubular fermenter. Make a note. If you know more points, you can add those points also. But kindly be familiar with all these spotters. So, which one is this? Hope you are able to identify here. This is fluidized bed reactor or bioreactor. Fluidized bed bioreactor. Description about the fluidized bed reactor. Take screenshot or pause the video and go through this answer. Next one. Which fermenter is this? Yes, you are correct. Drum fermenter, right? This is a description about drum fermenter. I don't know. Pause the video. Go through all these points. Okay, then. Which spotter is this? Yes, you are correct. Tray fermenter. Okay. And here you can see the description about tray fermenter. Yeah, it's given already. You need to recognize this as yogurt. Okay. And yogurt description is given here. You can have a look. Next, what is this? You can see here the algal slurry. Okay. So you must write here this as mass culture of algae spirulina. You can write, okay? You can write all these points, or in theory, whatever the explanation is given, no? Those points also you can write it here, okay? And also you can identify that as SEP, single cell protein. If you study all these things, no, this will be useful during your Viva. You can answer from these questions also. Then this is, what is this? Yes, you are correct. This is penicillin production. Okay, to identify this particular spotter, penicillin production, and this is the description about penicillin production. Hope I'm making myself clear to you. Hope you are able to recognize this spotter. What is this spotter? See the end product here, glutamic acid, right? So glutamic acid production. And this is the description about the glutamic acid production or production of glutamic acid. If I'm going very quick, kindly pause the video and go through this information, okay? Okay, which structure is this? Hope you're able to make out. Cobalt is present inside, right? This is vitamin B12 or cyanocobalamin, right? There's a huge structure you can keep in your mind. This is vitamin B12, and the respective description you can make a note here about the vitamin B12. Okay, so hope you're able to recognize this spotter, right? Which is this spotter? Yes, you can see here the grapes is there, no? So you can identify this as beer production. Okay, to identify that diagram or photograph as beer production. Before I conclude, let me go through all the spotters. Hope you're able to recall all the spotters. Let me check one by one. You also do the cross check so that you'll be perfect with this one. And only these spotters will be kept during your examination. Kindly focus on all these, okay? I'm going to keep only these spotters, whatever I'm showing right now to you, okay? Hope you're able to recall this one. This as still tank fermenter, okay? A typical fermenter, okay? Description. Then this is, yes, you're correct, airlift fermenter. This one, cheese. Say cheese. So this is packed bed bioreactor or packed bed fermenter. This one, tube, right? This is like a tube. So it's a tubular fermenter. What is this? You can see here the fluid, right? So you can say here as fluidized bed reactor. Okay, then you can see it's like a drum, right? So you can keep this in mind. Hint that's it, that can keep this as a hint. Okay, so this is, you can see here the quite cylindrical, half cylindrical can be seen here, right? So this is drum fermenter. Okay, the hint is it will be written first stage drum. So hint is drum fermenter, or it's this particular word you can catch, drum fermenter. See, second stage drum is here. Then the next one is, can identify this as tray fermenter. Here you can see like a tray, no? So this is tray fermenter. Okay, you are able to recognize this one. This is yogurt. Okay, hope you're able to recognize this one, right? Mass production of algae, that is spirulina. And also you can write that as SEP, single cell protein. So here the hint is penicillin, right? It is penicillium, chrysogenum is there, so slants. So you can identify this as penicillin production. The next one here, you can go through all this process, okay? And this is end product. The end product is glutamic acid. So you can write this as glutamic acid production, okay? And this one is, you can see this is a huge complicated structure. You can identify this as vitamin B12. This spotter will be kept. Okay, in previous examination, I had kept this particular spotter. So the same thing written here. So this is vitamin B12 and the respective description is given here, okay? So what is this? You can hope you're able to recall this one. This is beer production and this will be description. Hope you understood today's concept, and kindly study only those spotters more than enough, okay? And also every day take this affirmation, okay? I'm a warrior and I must fight against bad time. God helps those who helps themselves, okay? Or in general, if you want to keep this in your mind, you have to be a warrior. You must fight against bad time. God helps those who helps themselves, okay? And also keep in your mind that you should become a, or I should become a warrior instead of warrior. You should not become a warrior. You should become a warrior during all your circumstances. Okay. Thank you for watching this video and thank you for being with me. Kindly subscribe this channel to support. I, Dr. Pratibha K, signing off for today. Stay tuned with me, stay connected with me, stay safe. Thank you.